Our next group of pests that attack ornamental plants is a really large, diverse, and important group. While most folks think that all pests suck, the ones we will be covering literally do suck. Most of the sucking pests are hemipterans, and this order of insects is incredibly diverse in shapes, forms, feeding methods, and life cycles. In this module, I'll quickly present the groups taxonomically, but I want to spend most of the module discussing the nature of sucking pest attacks. Basically, there are three groups: cell content and tissue maceration feeders, phloem feeding feeders, and xylem feeding feeders. We've been using a hybrid of plant parts attacked, in other words, foliage, branches and trunks, roots, etc., and taxonomy, like the borers. Now I'll continue using taxonomic groups to discuss the sucking pests. The three main groups are the bugs and bug-like insects that are now in one order, the Hemiptera. I actually prefer the old taxonomic arrangement where the true bugs were in the Hemiptera and the bug-like insects were in the Homoptera. Genetic analysis has determined that all these sucking insects belong to one order, but there are three suborders: the Heteroptera, the Echinorinca, and the Stenorhyncha. Many of the Stenorhynchus bugs, like whiteflies and scales, have highly modified life stages which complicate their management. Remember that the term thrips is both singular and plural. These insects also have highly modified life cycles with pupil-like stages, though they are considered to be insects with incomplete life cycles. Mites technically don't have sucking mouthparts like the insects, but their tiny chelicerate mouthparts act as stylets to suck out plant cell contents. Sucking pests are often small and easily missed, but their damage can be very useful diagnostic aids. Sucking pests can cause plant distortions when they feed on juvenile tissues. Cells are damaged and killed, and as the leaves and stems continue to expand, the damaged tissues act as scar tissue, which is distorted and can distort further growth. Other sucking pests dissolve and macerate tissues within leaves or stems, and this results in dead spots, even on expanding plant parts. Salivary secretions can clog vascular bundles, thereby stunting, discoloring, or killing plant parts outward from the clogged area. Because sucking pests remove lots of liquids and little nutrients, they produce two types of diagnostic excrement: tar spots and honeydew. Tar spots are dark, shiny spots of resinous-like material. Honeydew is a clear, watery excrement that is produced mainly by insects that feed on plant phloem or xylem bundles. Phloem has lots of water and sugars, but little amino acids for proteins, so this excess is excreted. Xylem has even less nutrients. Honeydew often promotes the growth of sooty molds. Cell-feeding often produces tiny spots or speckles of yellow. When extensive, this can cause a general bronzing of or browning of the foliage. As previously stated, sucking insects commonly damage plant tissues that are still expanding. Twisting and curling of the foliage is a common result of feeding on expanding leaves, needles, and shoots. In the image with the leaf bug, you can see the honey locust leaflet has curled around the leaf bug feeding spot that is now turned brown. Below this are ash leaves that have been severely twisted by aphids feeding within the twisted foliage. In this case, the aphids are feeding in the phloem bundles that cause adjacent cells to distort. The fig tree with folded leaves is the result of thrips feeding as the leaves expand, not a leaf-folding caterpillar. The thrips first feed on tissues along the leaf midvein that causes the leaf to fold over. Finally, we see what is called hopper burn on red maple. The leafhoppers clog the xylem bundles, which cause the leaves to dwarf, curl, and discolor as if they had been scorched by fire. Sucking insects tend to take in a lot of fluid, so they need to get rid of the excess water. The insects and mites that feed on cell contents have a more concentrated food, so their excrement usually looks like a brown or black resin-like material. This is generally called tar spot. Plant bugs, lace bugs, thrips, and spider mites often leave behind tar spots where they have been feeding. Insects that feed on phloem and xylem fluids have to take in a lot of water, which they then need to extract to remove the sugars and amino acids. Phloem especially is so sugar-rich, the insects also excrete the excess sugars. Their watery, sticky excrement is called honeydew. Unfortunately, you will often see honeydew being called a secretion. Remember that secretions are materials produced by glands, so honeydew is an excretion. Honeydew can be deposited so heavily that it will collect on sidewalks, decks, park benches, and cars that are under infested trees and shrubs. I often chuckle when I hear that trees are dropping sap on cars. That would be a pretty stupid plant to go through the process of photosynthesis, then spit out the sugar. Actually, the deposits on the car is tree sap, but it's tree sap that's gone through the digestive tract of an insect. Sooty molds are a group of molds that feed on sugars. These fungi are found everywhere there are sucking insects. There are several genera and many species of fungi that can be called sooty molds. These fungi grow on sugars, some of which may actually be produced by the plants and occur on ripe fruit surfaces. The honeydew produced by insects is also used. When honeydew lands on a plant surface, the sooty mold fungi grow a mycelial mat, which is usually gray or black in color. Since the fungi do not penetrate the plant tissues, it can be scraped off, or it will eventually be weathered off once honeydew production stops. One group utilizes the copious honeydew produced by the beech blight aphid, and the mycelium can grow to an inch thick or more. Sooty molds also don't care about the substrate on which the honeydew lands. You may not have noticed that sidewalks are often gray or sooty under trees and shrubs. This is because honeydew-producing insects have been feeding on the overhanging plants and dropping their honeydew on the sidewalks. Sucking insects can cause what appears to be overall chlorosis, that is, yellowing of the foliage or spots of chlorosis. Euonymus scales usually attach to the undersurfaces of leaves, and their feeding in leaf cells causes yellow spots on the upper surface of the leaf. Plant bugs macerate little patches of leaf tissues, then extract the juices. This produces darkened, sunken spots on the leaves. Unfortunately, when this is done to an expanding leaf, the damaged spots may drop out, which makes the leaf look like it has been attacked by a skeletonizer or pit-feeding insect. Mites extract the contents of individual cells, which results in tiny yellow speckles. When mites feed on a lot of cells of a leaf or needle, the dead cells turn brown, and this gives the plant an overall bronze look. The hemipterans in general have modified their mandibles and maxillae by being extremely elongated so that they form piercing stylets. These stylets join together to form a food canal and a salivary canal. The bases of these stylets still have their muscle attachments to the head capsule so that they can slide back and forth to pierce plant tissues. The labrum is usually reduced or absent, but it may form a cap over the front of the stylets. Usually the labium forms a thick sheath that surrounds the stylets. When the insect feeds by pushing the stylets into the plant, the labium folds out of the way. Snodgrass was a famous insect morphologist who wrote the first major textbook on insect and arthropod morphology. I've captured the illustrations he modified from several other works of his time. Only the first two images show the labium, and in the case of the aphid, the labrum. The other two images don't show the labial sheath. In all of these, you can see the general trend of having the maxilla form the food and salivary canals. These are surrounded by the mandibles in various configurations. The first image is of a cicada, the second one is of an aphid, the third one is of the squash bug, and the fourth image is of a bedbug. This PowerPoint page came from a recent electron microscope study of the mouthparts of the spotted wing lanternfly that has invaded Pennsylvania. The author made a diagram of a cross-section of only the maxillary and mandibular stylets about halfway down the mouthpart. The large labium has been removed. I wanted to have you see the highly modified locking grooves and ridges that hold the maxilla together. What is amazing to me is that these stylets are still able to move back and forth in order to thread the stylets into plant stems where the stylets find and penetrate xylem bundles. Insects that feed on phloem fluids do the same with their stylets. There are chemoreceptors on the tips of these stylets that help the insect find the correct location for feeding. When I discussed the borders, I talked about the phloem, cambium, and xylem in trees and woody shrubs. Herbaceous plants still have the three types of tissues, but they can be configured quite differently than what we saw in trees. In many plants, the phloem, cambium, and xylem are located in bundles around the main stem and in leaf veins. This is important to understand as hemipterous plant-feeding insects are generally phloem or xylem feeders. Most of the bug-like insects feed in the phloem, but there are some plant hoppers and leaf hoppers as well as some scales that are xylem feeders. You are probably wondering why this is important to entomologists, and it has to do a lot with disease transmission and control of these insects with systemic insecticides. Most of the translocated systemic insecticides travel in the xylem, so phloem-feeding insects can occasionally avoid the toxins. Systemic insecticides that have higher water solubility seem to be able to move from the xylem up the plant, but then can diffuse into other tissues and kill those phloem-feeding pests. I also captured this PowerPoint page from the BioNinja website. Their site is actually dedicated to some laboratory techniques for obtaining pure phloem fluids out of plants. Basically, they let an aphid thread its stylets into the plant to locate the phloem bundles. Once the aphid begins to produce honeydew, they snip the mouthparts, and the phloem fluids continue to be pushed out of the attached stylets by the plant. Cool technique. However, what I want you to also see is that the diagram on the left that shows how an aphid threads its stylets through the various leaf cell layers until it finds the phloem bundle. The image to the far right is an actual microphotograph of a cross-section where an aphid has inserted its stylets. There are also some cases where a phloem tube can become clogged or damaged, and the insect is able to pull the stylets back and locate another phloem bundle nearby. Insects that take in fluids and move from plant to plant always have the potential of vectoring diseases. In the case of sucking pests, they are extremely efficient vectors of a host of virus diseases. Most of these diseases are important in vegetable production, but some of the viruses, like aster yellows, can severely affect ornamental perennials. Elm yellows is a major mycoplasma that kills elms, and it is vectored by leafhoppers. There are also quite a few xylem-infecting bacteria that are transmitted by leafhoppers. These bacteria often cause scorch problems in trees. Before we go through the Sucking Pest modules, let me quickly review some of the larger taxonomic groupings of the Hemiptera, the bugs and bug-like insects. The first suborder of this order is the Heteroptera, or true bugs. These are easily recognized by the presence of a triangular scutellum, and when the wings are folded, the wing membranes form a rhombus shape. When the front wings are pulled out, they have a thickened base and a membranous tip. The hind wings are entirely membranous. True bugs have elongate, segmented antennae. Most importantly, the mouthparts arise from the front of the head and are folded down the mid-ventral line between the legs. The Echinarhyncha suborder contains some of the bug-like insects that have the front wings all the same texture throughout. Those front wings may be thickened or membranous, and the hind wings are entirely membranous. These insects have bristle-like antennae, and their mouthparts appear to be attached to the back of the head, with the stylets arising just before the front of the leg bases. The group includes the cicadas, a host of jumping insects like leafhoppers, tree hoppers, plant hoppers, and spittlebugs. All feed on plant juices and are usually quite mobile. The Stenarhyncha are considered to be less mobile. These also have the front wings being the same texture throughout. Most have membranous front wings, but some have the front wings slightly thickened. The antennae are easily visible and consist of obvious segments. The mouthparts have migrated even further backward on the head, and the stylets appear to be arising from between the bases of the front legs. Many in this group have complicated annual life cycles, especially the aphids. Some also form galls, which we'll cover later on. The whiteflies and scales have modifications of their life cycle stages, in which they have mobile and sessile stages, and even go through a transformation stage that is commonly called a pupa, though it is not the same pupa as found in the true holometabolous insects.